Thank you everybody for joining us for the Plant Phenome webinar for February. I apologize, I made a mistake in the time conversion despite sending it out multiple times. I was thinking it was at 4 o'clock, and it's my fault we're late, so I apologize. But thankfully William is at 8 o'clock in Australian time, so he's well rested and ready to go. William Salter, Dr. William Salter, grew up in Scotland in 2012, and he moved to Australia to conduct his PhD in ecophysiology at the University of Sydney. He holds a postdoctoral research program with Professor Margaret Barber in the School of Life and Environmental Sciences at the Sydney Institute of Agriculture at the University of Sydney. He's also the social media editor for the open access journal AOB Plants, and is part of the social media and communications team of the Australian Society for Plant Scientists. He is a plant ecophysiologist interested in identifying plant phenotypic traits that could be useful for future food security and ecosystem resilience under climate change. He's worked with native Australian plants as well as important crop species. He's also interested in the development of new scientific tools and techniques to understand plant traits that have been overlooked in the past. He recently had a journal article published in the Plant Phenome Journal, which is why he was invited here. So I guess without any further ado, again, I apologize for the late start here, but Dr. William Salter will be talking about time-dependent bias in instantaneous ceptometry caused by row orientation. Thank you.
Yeah, thanks, Seth. Yeah, fitting that I have time in the title of my talk, because it's 8 a.m. here and I've got a coffee, so I'm ready to go. As Seth said, we recently had a paper published in the Plant Phenome Journal. I've just put up the reference to it here on my first slide. So if you want to go and have a look at that for some further reading after this talk, please go ahead. It's open access, as all of the Plant Phenome Journal ones are. So yeah, go ahead and have a read of that. My talk today, I will first give a background to this work, just to put it into the context of our larger project, which we have running here. Then I'll talk a bit about our parbar ceptometers, how to build them, and sort of how they compare to other research tools of this nature. I'll then talk about the results of this paper, and then go on to discuss the conclusions and the implications of this work. So first off, this paper came out of a much larger project. We have an international wheat yield partnership project. The IWYP is a CIMMYT-run program looking to increase yield potential by looking at traits that maybe haven't been looked at in the past. Our project, we were looking to increase carbon capture of wheat by optimizing whole canopy resource distribution. And so really there's two ways that we could approach this. We could increase the carbon capture by just increasing leaf photosynthesis per unit input, or we could distribute existing inputs more efficiently throughout the plant. And yeah, so in this project, we're focusing on the second of those two. And as I mentioned, this has often been overlooked in pre-breeding research in the past. But I mean, it's really important. Whole canopy photosynthesis is substantially reduced by the inefficient distribution of photosynthetic resources relative to the light that's available at different levels in the canopy. And so, yeah, just to sort of give an example, some people can sort of see it a bit more easily this way. On the left here, we have what we would ideally like all wheat to be. The A signifies photosynthetic resources, and the sun is the light level at the different levels in the canopy. So as you can see, we have quite a lot of light at the top of the canopy and lots of photosynthetic resources. As we move down the plant, we have less light, and we should, in theory, have less photosynthetic resources. However, as you can see on the right here, we generally don't see that in the field. We normally would see too little photosynthetic resources at the top relative to the light available, and too much at the bottom. My co-author on our ceptometry paper, Tom Buckley, he's at UC Davis, published this paper in 2013 in Plant, Cell and Environment. And as you can see on the left here, if we had an optimal distribution of photosynthetic resources, we've got photosynthetic capacity per unit daily irradiance on the y-axis here, and the daily irradiance on the x. As you can see, we get this nice curve. So as you get more light, you will have more photosynthetic resources per unit irradiance. But in actual fact, what we see in the field is almost the exact opposite of this. We have way too many photosynthetic resources in the lower canopy where there's less light, and far too little in the top where we have much more light. And really what this means, if we could shift from this really suboptimal distribution of resources to this nice optimal distribution, then we could get increases of about 20% in daily carbon gain. And this percentage sunshine, so direct sunlight penetrates less into the canopy, whereas more diffuse light penetrates deeper into the canopy. So that's why we would see a larger increase if we had more direct sunlight than diffuse. But regardless, across the board, we would get increases in carbon capture. But you're asking, how do we actually achieve this? Well, look, we set out to screen 160 genotypes of field-grown wheat for both photosynthetic capacity in flag and second leaves, and also the daily light interception by these leaves. From that, we'll put them together to look at how optimally the photosynthetic resources are distributed with regards to the light. And from that, we can do some genomic analysis like GWAS. And look, at the end of the day, we want to use these to breed for more efficient crops and increase the yield potential of wheat. That's what IWYP is all about. But with the phenotyping tools, sort of standard plant physiology phenotyping tools, how do we achieve this with adequate replication? Well, I'm not going to talk about the photosynthetic capacity today. I have added a couple of slides at the end, so if I have time, I'll talk a bit about that. But really, I'm going to talk about how we measured the daily light interception by the flag and second leaves today. And so we did think that ceptometry was the answer, but we wanted to maybe use something different from standard ceptometry practice. And so I'll just give a brief overview of how people normally use ceptometry, and then go over the flaws, and then how we sort of quantified one of these major flaws. So standard ceptometer practice, this up the top here is a ceptometer that probably most plant physiology labs around the world have in their supplies. And basically what we have is a light bar, which has light sensors underneath a diffuser. That's what you put into the canopy, and then you have a small quantum sensor which you hold above the canopy. And so with this device, we get a measurement of photosynthetically active radiation both above the canopy and below it. And from those two measurements, we can estimate canopy light interception and biomass traits such as plant area index or leaf area index. And as I mentioned, these tools are sort of everywhere, and they're widely used in the research of agricultural crops. How do we use these? Well, we have our little field plot here, and our little Pac-Man with a ceptometer in his hand. Generally, what people will do is they will start at about 11 o'clock in the morning, and they will start walking around their field plots, taking measurement in one plot after the other. And yeah, as I mentioned, these measurements are normally taken between 11 and 2 in the afternoon, and that's as per CIMIC guidelines. And the reason for that is, as this little Pac-Man is walking around the field, time is ticking away, and the sun is also moving across the sky. And so that's not really a good thing, but we can correct for certain factors. So we can correct the canopy transmittance. This is prior to getting an estimate of leaf area index or plant area index. We can correct those measurements of canopy transmittance for the beam fraction of incoming power, the leaf absorbance, which can differ between different crop species, the canopy extinction coefficient, and the main thing that affects that is the solar zenith angle, and we can correct for that using based on the location you're at and the time of day, and also the leaf angle distribution, which again can change with different crop species. However, the biggest flaw in this method is that clumping can bias the estimates of transmission, tau here, and the estimates of, and therefore the estimates of plant area index. And this is particularly significant for row crops, where of course you've got big gaps in the canopy between the rows of planting. And so the light interception of the ceptometer, which you place below the canopy, will ultimately depend on both the solar zenith angle and the difference in the row planting angle and the interaction with the solar zenith angle. And like this has been known about for a very long time. Modeling papers in the 1980s brought this up. However, it's largely been overlooked for some reason, even though it was known about. And it's not really been quantified in the field. So we decided we sort of had to do this. And like the other flaws in taking just spot measurements of canopy light transmission is that you miss variation in the canopy light environment over time. And this has been highlighted quite a bit recently. If you want to read more about sort of quick changes in the light environment and how it affects plant physiology, then there's a few nice reviews. There's this paper by Slattery et al., which was published last year in Plant Physiology. So if you think about a canopy, the canopy can move, and that will allow direct sunlight to penetrate to deeper canopy levels. And you also have clouds passing over. What we call these short bursts of light to the lower canopy are sunflex. And they vary in length and time, and there's a lot of research going on in that as well. But really, taking instant measurements, you're missing all of this information. And it is very important for photosynthesis and other physiological properties and processes. As you can see, photosynthesis takes about 20 minutes to sort of ramp up to full speed. That's due to enzyme activation. It's also down to the stomata opening and also photoprotection relaxing as a plant is after it's been exposed to full sunlight. So yeah, with your instant measurements, you're missing all of that. And you're also missing over sort of longer term, you're missing how the light environment develops as a crop develops. And so this here is from a recent preprint that we put out, showing how the plant area index increases as the crop develops. I mean, that's sort of what we would expect. Up the top here, we've got a bit of senescence occurring, and so it begins to drop off after this point. Again, spot measurements, you're not really going to capture that. But the big problem with not taking spot measurements and taking, let's say, continuous measurements across whole days is that these ceptometers, like the one I showed earlier, Costs on the order of sort of $2,000 plus. So you're really not going to be able to put lots of ceptometers out into all your various research plots at the same time and measure across a whole diurnal cycle. So that's why we decided to build our own. And so the aims of this study were twofold. We first wanted to demonstrate that we could build high accuracy, continuously logging ceptometers that we could just put out into the field for whole days or whole seasons, as I just showed in that last graph. And also to assess the impact of diurnal variations in the solar alignment with row planting on estimates of plant area index estimated from instantaneous measurements of ceptometry. So let's, without further ado, I'll introduce our parbar ceptometers. So this was our solution that we came up with. It works in much the same way as that other ceptometer that I showed earlier. It gives us a simple voltage output, so that's an integrated measurement across a meter length. And so we can put that under the canopy and we get one light measurement for the whole canopy there. The good thing is, because of the simplicity of the output, it's compatible with almost every data logger out there. We also made it weatherproof, so we can just throw it out in the field and just leave it there. It's also very easy to build. What we have here is just an aluminium U-shaped bar and then an acrylic diffuser along the top. On the underside of this diffuser, we have 50 photodiodes, which we just solder to 2 copper wires, and then just attach a waterproof DC connector to the end. And then we linearize the whole response to power using just a temperature stable precision resistor, just connected in parallel with the circuit. So, I mean, really, it looks quite complicated, but in actual fact, it's very easy to build. All you really need is some basic soldering skills and some DIY know-how. To put it into perspective, I actually had never touched a soldering iron before I started this postdoc project, and now we have about 100 of these sitting in the field store just down from my office. The other good thing is that they can be built in batches, and the best thing about them is that in parts they only cost about $75 each. That's compared to the sort of $2,000 that I mentioned earlier. And we don't actually sacrifice any data quality. This is a calibration curve of one of our power bars. And as you can see, it's actually very, very good. We have an R² value of, I mean, pretty much 1, as close to 1 as you're going to get. And we calibrated this against a commercially available quantum sensor, which costs in itself over $500. So if you're saying, well, $75, it's not going to be as good as a device that costs $2,000. Well, I think this graph just proves that that's a completely wrong call there. As I said, it's weatherproof, so we can put it out in the field in rain or shine or hail or snow, whatever you want, it's going to work. So yeah, we're planning to put some out in the field for a whole season next year. Getting on to how we actually conducted this study. So we had field plots up at Narrabri in New South Wales in Australia. I guess the big thing to note with this particular study and our results is that we had a northwest to southeast row orientation. And that might not be normal for a lot of research plots. They'd probably normally be oriented sort of north to south. But for farmers and other sort of people who are growing crops, they could be oriented in any sort of direction. For this study, we actually looked at 239 genotypes of wheat measured at Anphesis, and as I said, we didn't stray too far from sort of normal ceptometry practice in terms of what we did. It was just that we put them out there to measure across the whole day. We had a quantum sensor above the canopy, and we put the power bars below the canopy. And we used a Campbell scientific data logger to take measurements every 10 seconds throughout the day. What we found, well, we were quite surprised by how large the effect of row orientation was on these on our measurements of transmittance. We found it to vary hugely across the day, despite constant canopy architecture. I mean, these plants weren't changing how their leaves were oriented across the day, but we found this big sort of spike in the morning. And a big peak of transmission in the afternoon as well. And in the middle of the day, there was a drop in transmission. And look, we suspect that afternoon peak is due to alignment of sun with the rows. As I said, we're in a northwest to southeast direction there. And in Australia, the sun's in the north of the sky. I always have to remember to say that, because it's not something everyone thinks about when we show these results. And then the morning, so in the afternoon, you've got the sun aligning up sort of quite well with the row orientation, and so that's allowing light to transmit to the bottom of the canopy where we've got our ceptometer. Likewise, in the morning, the sun will be sort of coming from a northeast direction to the southwest, and that will give us that possibly explains this peak in the morning as it's penetrating in between tillers perpendicular to the row planting direction. What does this mean for plant area index? Well, as I said, we correct for a number of factors during the estimation of plant area index. But even despite these corrections, we found that estimates of plant area index varied widely across the day. And so if we consider that you might be walking around your plots taking instantaneous measurements, let's say you've got hundreds of plots, it will take you 1 to 2 hours to walk around your whole plot. And as you can see, in the mid-morning down here, you would underestimate plant area index by approximately 28%. Whereas when you walk just about 2 hours along your plots, you'll actually be overestimating it by 32%. And the horizontal line here is our diurnal average plant area index, which we measured, we took an average across the whole day of these measurements here. And so, I mean, these are big numbers. If you're going to be making decisions about breeding based on plant area index or leaf area index, this is huge. And so, yeah, we feel this is absolutely massive for people who are using ceptometers in their daily research. As I said, instant measurements would be subject to time-dependent bias, which I showed in that last graph. And that's caused by the diurnal shifts that occur with the alignment of the solar beam with that row planting angle. Unfortunately, we don't think this can be resolved with stationary corrections, as we mentioned earlier, which you can just say you're in this location, because it's really quite crop and site specific. If we had taller plants, you would actually get the effect might change, and also if you had different leaves, if you had broadleaf species, that would also be different as well. It's also highly site-specific, and often the orientation of the rows is actually down to historic land use rather than anything else. And like, although I said our plots were in the orientation that we had up in Narrabri, we did have some plots out over in Davis as well. And these were planted in a north-south direction, and we still saw some variation across the day. It possibly wasn't quite as stark as this, but it was still there, and it was all down to this the sun moving across the sky and lining up with the rows at certain times. So what we would ideally recommend is that everyone was using continuously logging ceptometers in all of their plots. But this isn't going to be feasible for everyone. But we would recommend putting some out into some plots to develop site-specific correction functions for the crop species that you're wanting to measure. And that will look something like this. So here's our Pac-Man again. He's walking around his plots as he was before, but this time we have continuously logging ceptometers in some of these plots. And from these continuously logging ceptometers, we can develop a site-specific correction function, which can then be used to correct our Pac-Man's instantaneous measurements. When we take the data before it's corrected, we get these black lines here and these black bars. This in the bottom left is the PAI relative to the diurnal average PAI, and as you can see, without correcting our Pac-Man's instantaneous measurements, we get big overestimates of PAI at this time, and we're sort of hitting a diurnal average at that time. However, if we correct for this data with our correction function here, we get these red lines, which are much, much closer to the diurnal average. And likewise here, it's just the data presented in a different way. But as you can see, the PAI inferred from the instantaneous measurements is much more consistent across the day. Well, actually not across the day, across that recommended measurement period that CIMMYT recommend. I guess some final take-home messages from our ceptometry work is that we should be a bit more wary of instantaneous ceptometry inferred estimates of LAI and PAI. There's a huge amount of error there, this error and bias could be eliminated by using continuous measurements, or continuous measurements could be used to develop site-specific correction functions, as I showed on that previous slide. But I guess the biggest thing from this work is that you can build your own research-grade ceptometer for $75 with just some basic soldering and DIY skills. And we were so excited about this last point that we actually submitted a manuscript to Jove, which is a Journal of Visualized Experiments, and that just got accepted last week. So soon enough, we'll actually have a video protocol online showing you how to build ceptometers of your own. So yeah, we're quite excited about that one. I think I'm going okay for time, so I will just sort of go off on another tangent. But I am talking in the Plant Phenome Journal webinar series. I think it's still relevant to talk about the high throughput gas exchange system we use. As I mentioned earlier, in our project, we also need to measure the photosynthetic capacity of flag and secondaries. And like a Lycor 6400 or 6800 just wasn't going to cut it. It takes way too long for us to take measurements with that system. So instead, we designed our own, and this is a multiplex gas exchange system, which gives us 8 leaf chambers. We still only have one infrared gas analyzer to analyze the gas that's going through each chamber, but we have a solenoid switching manifold that allows us to switch through the gas coming from each chamber. And I mean, it's a fairly simple design. When it comes to gas exchange systems, we can't do things like ACI curves or light response curves, but we can take lots and lots of measurements of photosynthetic capacity, approximately 20 measurements of photosynthetic capacity per hour. And what does it look like we could do across the whole day? Well, this is just one day's worth of data. We've got photosynthetic capacity on the y-axis and then just the individual leaves on the x. And so, I mean, really, this allowed us to measure these 160 genotypes, and we're super happy with it. And we recently published our methods paper on that system in Plant Methods. Again, this is open access, so please go and have a look. We're super happy with our OCTOflux system as well. And so we are working on a much larger paper combining the results of both combining the data from our power bars and our OCTOflux system. So keep an eye out for that as well. In our lab, we're also interested in sort of building other cheap instruments that don't compromise on data quality. So we've built $2 quantum sensors using exactly the same photodiode as is in our ceptometers. Again, compared to a Lycor quantum sensor, we get a really good calibration curve. We're also building DIY sap flow probes, and Tom Buckley over in Davis is continuing to use these now in almond orchards, I think. And we're also looking to build very cheap data loggers using the Arduino platform. And we're sort of getting that up to speed now. We can now give 12-volt power to sensors and things like that. So yeah, it's all quite exciting. And we've built a couple of other gas exchange systems too. But with that, I'm going to stop. First, I'd like to thank my co-authors on this paper, Dr. Tom Buckley and Dr. Matthew Gilbert over at UC Davis. I'd also like to thank all of my colleagues at Sydney who are involved in this project, particularly Julie Lintz. She's actually back in France now, but she was helping us with all of this ceptometry work out in the field, moving the power bars from plot to plot each evening and things like that. So yeah, she was great too. And finally, I just need to thank our funders. So we're funded by the GRDC and in Australia and USAID in America, and we're also supported by IWYP. Finally, I just wanted to show this photo. So this is our ceptometers out in the field. I'm pretty happy with that photo. So with that, I'm happy to take any questions that you may have.
All right. Well, thank you, Dr. Salter. Getting a little feedback there, so I'm going to turn on my mic. So that was a really interesting and looks like you got a lot of new instruments that are going to lead to new generation of discoveries. So really exciting stuff. So we are going to open it up for 15 minutes or so for questions. And so you can write that in the chat box, either directed at myself or Dr. Salter, or just to everybody, and I'll read it. Or you can unmute and then just turn down your volume and introduce yourself and go ahead and ask your question. So I'll start with the first question here. It looks like a pretty sort of damning case for sort of those the status quo. So have you gone back and taken a look and seen how many I guess other authors have done it in a way that their conclusions would be erroneous at this point? I mean, are we talking about 10 studies or hundreds of studies, or?
I haven't actually gone back to look, but look, I'd probably go as far to say that almost every study that has used a ceptometer in row crops will have some bias in the measurements. I mean, it really depends on how many measurements they've taken across how many genotypes and things like that. But I mean, standard ceptometry practice says you should sort of minimize the amount of time that measurements are taken over. But yeah, I mean, even that, with those figures that I showed, you had a huge shift in the estimate of PAI even in just a couple of hours. So we think it's huge.
Great. Any people who would like to try and ask a question orally? I don't see anything in the chat box yet. Go ahead and unmute yourself if you want to do so. And I guess while we're waiting, I have a series of questions here. So on your you mentioned PAR, which is photosynthetically active radiation. There are other light measurements that aren't PAR. How can you talk a little bit about how those differ, and are all ceptometry actually measuring PAR, or do some use just regular light sensors?
Yeah, look, generally speaking, ceptometers will use PAR only. That is one of the reasons that ceptometers have been sort of exorbitantly expensive in the past, because the light sensors themselves haven't been specific to that PAR waveband, which, and simply speaking, is really just visible light. And so they've had to use expensive filters in their design. Look, we found this photodiode, which has very good spectral sensitivity just in that PAR waveband. So when you go down into the UV waveband, we get almost no signal at all. And likewise, if we go up into the infrared, we don't really get any signal either. It's highly specific to that visible waveband, which means that we don't need to use expensive filters or anything like that. So yeah, that's really the main thing that's allowed us to keep the cost down in the design of our sensors.
Oh, that's very helpful. I think I missed that detail there. So it's not.
I don't know if in the talk either. So.
Okay. Any other questions? Seeing none, let me ask you one other question. So UAVs, unmanned aerial vehicles.
Yep. I thought it might be coming.
Sorry?
I thought that question might be coming.
Oh, well, I guess I'm sort of curious how if they will replace some of these on-the-ground measurements. The nice part is they can get all the measurements done at the same time, but I guess they're also not under the canopy. So advantage.
I've actually just in the past couple of weeks tried to look at some data that we collected using stereo imaging. So that was actually on a sort of rover that went over the crops. And look, even with that, we found it very difficult to pick up all of the leaves in the canopy, especially when you've got leaves overlapping other leaves and things like that. I mean, there are other things such as LiDAR and things like that, but I mean, I guess what we feel is that all of these methods need to be properly validated against ground-based measurements such as ceptometry. We don't think that there isn't a place for drones, but possibly drones combined with ground-based measurements is the way to go at the moment. Yeah.
Okay, seems reasonable.